Well, I promised earlier in the semester I could ruin a lot of things for you, and now we've arrived at the chapter on digestive system infections. Fortunately, most of us are eating at home and can control food preparation, so here will be the chapter where we'll learn even more important ways to keep your food system and your digestive system safe. These are two electron micrographs. They're showing the curly non-producing cells, so they don't look curly, and these are curly producing cells. There are adhesive fibers that the E. coli produce that help them to stick to the intestinal epithelia. And this E. coli 0157H7 is found in the digestive tract of animals. We have E. coli in our digestive tract, but it is a different subspecies or a different strain of E. coli. The O and the H refer to parts of the lipopolysaccharide outer membrane of gram-negative, so the LPS. And it's the particular O antigen and H antigen combination. This one in particular can cause a hemorrhagic intestinal disease in humans if we ingest these bacteria, typically from undercooked ground beef, but it can also happen with produce that has been fertilized with cattle manure, and if those produce aren't washed properly, then that E. coli can get into our system. Once again, we'll have about a 3 or 4 slide complete anatomy and physiology of the system we're studying, just so that we know where there will be normal flora and where there should not be any normal flora. Obviously, in our upper digestive system of the mouth and the tongue and the throat and the upper pharynx, when our esophagus will have some microbes in it. But once we get down into the stomach and the liver and the accessory organs, there should be no microbiota in that part of our bodies. But when we get into the large intestines, we definitely have normal flora in that region. The entire digestive system is considered external because it is open on either end. Way back when we first started talking about the normal microbiota, or the normal flora, we said it is only on external surfaces. And our digestive system is certainly colonized with normal flora, and that is because it is technically outside of us. Looking in the mouth, our digestion, of course, starts as soon as we put food in our mouths. We use our teeth to grind it into smaller pieces, which increases the surface area. And that allows the enzymes in our saliva to begin breaking down the food into smaller and more chemically useful particles. So our salivary glands are accessory digestive organs because that's what's secreting the saliva and the enzymes that will not only moisten our food and make it easier to swallow, but also begin to digest it. Looking at teeth, you know, it's hard to think of it, but teeth are part of our digestive system because they are part of that grinding the food down into smaller particles. You don't need to know all the different parts of it, but when we look at the areas that can become diseased, the gums should definitely be touching the teeth and be secure against that surface. The enamel is the external surface, and we get deeper in layers until we get to the pulp, which actually does have blood vessels and nerves. And because of that, if we have dramatic tooth decay or breakage, dental disease can actually become systemic because it can enter into the bloodstream. That is why dental care is a critical part of our health care. Our small intestine has where a nutrient absorption occurs in the body, and we have lots of villi that increase the surface area, and there's significant blood flow to that, as well as our lymphatic systems. We saw goblet cells in our respiratory system because mucus is important there for trapping microbes and helping our ciliated cells to remove them from our respiratory system. In our digestive system, they produce the mucus that protects and lines our surfaces and also could trap any microbes that shouldn't be there and be allowed to get removed. Looking at tooth decay, that happens because of acid that is made during fermentation. So microbes on the surface, and particularly down near our gums where it's more anaerobic, biofilms form. That means on the surface, oxygen can get to those microbes. Below, the microbes that are deeper between the surface and the tooth are in an anaerobic environment. They carry out fermentation that produces acids. The acids degenerate the enamel and cause tooth decay. If it gets really deep, it goes beyond the enamel, down to the dentin, and even into the pulp, where we can get tooth abscesses and systemic infections. So when we go to the dentist, as we should twice a year, to get tartar removed, this tartar is built up to a significant degree where it becomes dental calculus. And the decay is starting in the crowns of the tooth. And this person, the decay has been drilled out, so there is no bacteria or rotten material left in that tooth, and then is filled with amalgam or that mercury-based fillings. Gingivitis, that mouthwash and brushing helps to prevent, is inflammation of the gums. The gingiva are our gums. So this person has buildup of tartar around the edges of their teeth, and that is causing inflammation of their gums. When the gums become inflamed, they can pull away from the enamel, and then that allows bacteria to get even deeper and cause problems. So the healthy gums shouldn't bleed, not even when you floss. In the early stages of periodontal disease, that's anything happening around the gums and teeth, perio around the teeth. So gingivitis is the first stage where the gums are inflamed. Periodontitis, we're getting decay of the enamel and degradation of the gum tissue. And advanced, we're getting significant tooth loss that could lead to systemic infections and even bone loss. This is an image of trench mouth. It's called that because it was first described, I'm sure it existed before, but it was first described in medical journals from World War I soldiers who were fighting in the trenches and did not have access to healthy diets or oral care and had necrotizing ulcerative gingivitis, meaning ulcers or sores are forming in the gums, necrotizing, it's killing off the tissue. This is a cold sore caused by herpes simplex virus 1 or HSV1. It can be on the lips, around the lips, it can even be inside the mouth. This is a virus that a significant portion of the population has, and many people will suffer from the outbreaks of these extremely painful blisters. It cannot be cured. Herpes viruses are in your body forever once you contract one, and they can be managed with acyclovir and other antiviral drugs that will decrease the frequency and the severity of a herpes outbreak, but won't cure the person from having that. I've also noted that oral herpes can also be caused by HSV2, herpes simplex virus 2. That would typically be transmitted from a case of genital herpes to the mouth via oral sex. And in the second picture, it's acute herpetic gingivostomatitis, where the whole back of the mouth is inflamed with the herpes virus. This is a picture of oral thrush, which is a yeast infection. We saw that in the genitourinary system diseases, because it is a common vaginal infection. But if it's in the mouth, it's called oral thrush. The white patches are the overgrowth and colonies of the yeast in the mouth. It is a fungal infection, and it is typically caused an oral thrush by suppressed immunity or overuse of antibiotics or long-term use of antibiotics that are necessary for a person. The thrush can be a part of our normal flora, but if that is disrupted and the bacteria are reduced in numbers, then the yeast overgrow. Mumps is actually a digestive system disease. I don't think I knew that until I started teaching microbiology. It's a digestive system disease because it is the salivary glands, the parotid salivary glands, that get inflamed and causes this severe swelling. It's very painful. Side effects can be deafness because the glands are right up in here, can press on the ear canal and inflame the eustachian tubes and cause deafness. In teens and adults can cause orchitis. In persons with testes, it causes inflammation and can lead to sterility. Prevention would be good hygiene, but primarily vaccination. It is part of the MMR vaccine, which is measles, mumps, and rubella. We looked at the measles and the rubella in a different chapter, and then the mumps is in this chapter because it affects the salivary glands. I have noted here that to see the tables, I put them in the Canvas module with this chapter. It is titled the Chapter 24 Study Guide, where I put the actual diseases that you will need to know for exam 4. It's a similar table to this, where I will list the diseases, and you need to refer to the tables to find the pathogen and the rest of the information. If there is more than one pathogen, you only need to know one. Likewise, if there's more than one drug that treats it, you only need to know one. So as long as you know at least one example from each of the columns, you will be in excellent shape. This thermometer has a big red star with a danger zone because that is the temperature that is optimum for microbial growth of human pathogens. Any organism that can cause disease in us has that danger zone optimum temperature because it is our body temperature. Our body temperature is around 37 degrees centigrade, which is 98.6 is the average temperature that they list. I used to hover at 97.5. In later life, I'm now at 98. If I hit 98.6 or higher, I actually have a fever. But these are the mesophiles. Human pathogens are all mesophiles, medium lovers, because their enzymes work optimally at the same temperature as our enzymes work optimally. So if you cool things down to refrigerator or freezer temperature, you're not killing the microbe, but you are slowing their metabolism down enough that there aren't going to be very many microbes present. And if that is their preferred temperature and they are replicating, getting into us will mean they're no longer at their optimum temperature and they won't survive. Hotter than that, we certainly can't tolerate these, but 60 degrees centigrade or 140 Fahrenheit is the holding temperature for cooked foods. So if you cook a food and then hold it at least 140, that will prevent microbial growth. Then we have different temperatures listed for the temperature that you have to cook foods to get them to that safe temperature before you can let them go as low as 140 for the holding temperature. This is an image of Salmonella entering an intestinal epithelial cell. It's actually kind of a pretty image. So the ruffled green is the human epithelial cell. You can see that there's a lot of twists and curves to increase that surface area for nutrient absorption. And these bacteria are bacillus shaped. They have these long extensions that are pili that help them to attach, and then they reorganize the host cytoskeleton, those protein fibers inside the cells that give it its shape and hold organelles in place. The bacteria can secrete chemicals to reorganize that cytoskeleton that tricks the cell into engulfing the bacteria and bringing it in, not to be phagocytized and eliminated, but to be in a vacuole where it can now replicate and then spread inside the host cells. Images of cholera. Cholera can cause fluid losses up to 20 liters per day. And you think, well, I don't have 20 liters of fluid. Yeah, exactly. You keep replacing it and it keeps exiting. And it does have up to a 50% mortality if the water and electrolytes cannot be maintained inside the patient. It secretes a toxin that causes chloride ion loss, and when the chloride leaves, so does the water. Typically found in places with poor sanitation, people who are unfortunate and do not have access to clean running water, where whatever water they have is the source for drinking and washing dishes and doing laundry and getting rid of human waste. It's all one place. It is fecal to oral contamination or transmission. And of course you think, but I don't eat poop. Yeah, you probably do. Contaminated water we probably would be able to stay away from and filter it, but other people cannot. One of the more striking examples of a recent cholera outbreak that I know of happened in Haiti after a hurricane. The UN troops came in to give aid, building cleaner water systems and helping set up hospitals, etc. But they had an improperly dug latrine, and one or more of the helpers was a carrier of cholera bacteria that contaminated the system. Haiti had never ever seen cholera before, so the mortality rate was astounding there. Not exactly happier, but less traumatic, is peptic ulcers. Peptic meaning it happens in the stomach. Ulcer is a sore or a hole. Helicobacter infection is what causes ulcers. For the longest time it was believed that stress is what caused ulcers because your corticosteroids go up. The thought was that was increasing acid production in the stomach, and that is what caused the ulcers. And that might contribute, but the actual causative agent is this microbe, Helicobacter pylori. Helicobacter because it spins like a little helicopter. It has the capacity to dissolve the mucus that's secreted by those goblet cells. When it does so, it's making an indentation. Then it generates ammonia that neutralizes the stomach acid that provides a more hospitable environment for the microbe to grow and keep spreading. And when it causes this damage, if it keeps digging down through the tissues, it can actually get to the blood vessels and cause a bleeding ulcer. C. diff is the abbreviation for Clostridium difficile, which Clostridium are all gram-positive, and they're strict anaerobes. And difficile is the Latin word for difficult. It is difficult to get rid of. It typically happens in the colon when the normal microbiota has been disrupted. That happens from people who, for various reasons, have actually required repeated courses of antibiotics. That disrupts the balance of the microbiota in the colon, and the Clostridium difficile, or C. diff, multiplies. This organism makes toxins that cause an immune response. The toxins are antigens. When the immune response kicks in, it causes inflammation. The monocytes migrate to the site of infection. The inflammation and dead cells form a pseudomembrane, and you get these very odd pockets formed in the colon and can actually be life-threatening. The treatment for this is a fecal transplant, where feces from typically a related person, just because you have normal related MHCs. This isn't a transplant where you have an immune rejection, but If you are related to someone, your life experiences, your environment where you grew up is going to be similar. Your microbiota will be similar, your normal microbiota. So having someone related will have closer to what should be your normal microbiota, where they can insert the fecal matter and reestablish a normal balance. And again, see that chapter 24 study guide. It doesn't list every single organism here. There will be a shorter list of them. It's still significant. I recognize that. And again, you only need to know one organism, a couple of signs and symptoms, one mechanism of transmission, and one treatment. Moving on to viruses, this is a rotavirus, and it is from a fecal sample. So once again, fecal to oral transmission, lack of hand hygiene. I'm hopeful that if nothing else, this pandemic will have taught us all the importance of hand washing, not just to prevent coronavirus, but from pretty much anything else that could get into our respiratory digestive system. This is what's typically called the stomach flu. It is not the flu. It's not caused by influenza, but it does affect the stomach and have vomiting and diarrhea, cramping, body aches. And we have the norovirus and rotavirus, both very similar transmission and symptoms. There is a vaccine for infants for the rotavirus. There is no vaccine for the norovirus. There are 5 different types of hepatitis. They're all called hepatitis because it's inflammation of the liver. Hepatitis A and B both have vaccines that can prevent them. Hepatitis A is typically a fecal to oral route transmission, contaminated water, food preparers having dirty hands, not having washed from using the bathroom and then preparing food or drinks can transmit it. Hepatitis B it's a DNA envelope virus, and hepatitis C is also an envelope virus. They have different types of genomes. Hepatitis A is a single-stranded RNA virus. Hepatitis B is a hepadnavirus, or HEPA, which is liver DNA virus. Hepatitis C is a single-stranded RNA, but positive. D is single-stranded and negative. And E is single-stranded RNA and positive. So it's not their genome that classifies them as a hepatitis virus. It's a fact that they affect the liver. So hepatitis, the symptoms, causes bilirubin from damaged liver cells, causes this yellowing of the skin and the whites of the eyes. It's called jaundice. And let's see, we have the contaminated food for hepatitis A. Hepatitis B, contact with affected body fluids. And hepatitis C, infected body fluids. Hepatitis D, body fluids, but only happens in people who already have hepatitis B. So if you don't have hep B, you can't get hep D. And hepatitis E is from fecal to oral route mostly in developing countries that don't have access to clean water. Even though the transmission might not be through the digestive system, it affects the liver, and that's why it's in the digestive system chapter. Cryptosporidium is there we go, is a protist. That means it is a single-celled eukaryotic microbe with transmission the fecal to oral route. It is a very large cell. We see the size band here is 10 microns. Cyclospora is also a protist. It also has fecal to oral transmission. So both of these protists transmitted by contaminated water. Also part of the study guide tables. Now we're moving into larger eukaryotes. the helminths, which would be worms. Here we see Ascaris. So the adult worms can be big enough to cause intestinal blockage. They will come out the system through excretion in the fecal matter. And in image C, we're seeing a fertilized egg, and that can be ingested, and when it becomes goes through mitosis to reproduce and create the organism, it's in your intestines, and that's where it can cause intestinal blockages and it can absorb your nutrients. Hookworm called that because it looks like a hook, and it does attach to the intestinal wall, and it can be seen on the skin surface when they migrate outside the digestive system. And it is obtained from contaminated drinking water. Nematodes are roundworms. These are called pinworms because they are very pointy on either end. Image B is the eggs, and that is most likely what would be ingested from the contaminated water, and then they would grow and develop into the actual worms inside the body. These are whipworms. They get ingested as the egg form and go to the intestines, and this is an unfortunate child that is suffering from rectal prolapse. From the infection, we lose the musculature that holds it into the body, and then it protrudes. These are tapeworms. The T stands for tinea, T-I-N-E-A. So tinea spiralis because it makes these swirled formations in the muscle tissues. So we would contract this from eating undercooked meat. This is a light micrograph, the suckers on the head of the tapeworm and the attachment, the spiky attachment that it would use to stick to your intestinal lining. And it's the eggs of these worms are also a way that we could contract it by ingesting water contaminated with those eggs. I believe lastly we have liver flukes. They're called that because they look like little livers. They are flatworms, and they infect the intestines. They have suckers that adhere them to our intestinal walls. And all of these worms can be treated with drugs, but the drugs are pretty harsh because worms are multicellular eukaryotes. We are multicellular eukaryotes. There are very few cell structures that a worm has that we do not. So there's not a lot of targets that are very specific to that parasite that won't also cause harm to us. So again, the actual list to know will be on the Chapter 24 study guide. There is still a lot, but that's why I started putting these charts in with the chapter as we go, as opposed to waiting a week before the exam.